Welcome back to this next video in which we are discussing influenza. So we've now discussed the three species of influenza virus, the influenza A virus, the influenza B virus, and the influenza C virus. And we're now ready to begin story time. We're ready to begin the story of the pathogenesis of influenza. Okay, so the first thing to say is that this story is pretty much the same for all three different species of influenza virus. There are slight modifications, slight differences between the different species of influenza virus, however they're minor, and when we see them, I will point them out. But for the most part, when I'm describing this story, I will not specify which species of influenza virus we are talking about, and the reason is that it will apply for all three of the different species of influenza virus. The story is general for all of them. Where there is a bit that is different between the different species, I will point that out and tell you how it is different between the different species. Okay, so let's actually begin pathogenesis. So, to begin with then, we need to discuss transmission. How, if you've got one unhealthy person with influenza, and you've got one healthy person who doesn't have influenza, does it transmit from the unhealthy person to the healthy person? Well, influenza is a classical example of the saying, coughs and sneezes spread diseases. And it's utterly true in the case of influenza. When the unhealthy person coughs in an effort to clear the airways, so the bronchi and the trachea, and the larynx, of course, and when the unhealthy person sneezes, which of course is to try and clear the nasal cavity, loads and loads of the virus particles will be expelled into the surrounding air, and then the healthy person can breathe those in, and then they have the potential to then get the disease as well. Now, of those two things, coughing and sneezing, which is worse? It's sneezing. Sneezing is much, much worse. The number of virus particles that are expelled from the nasal cavity when you sneeze is far greater than the number of virus particles that are expelled from the airways when you cough. So sneezing is the worst one for passing on diseases to other people than coughing. Okay, the other thing to say is that it's common knowledge that influenza is much more prevalent in the winter months. Now, there are loads of different hypotheses as to why this is the case. In my opinion, and in many other people's opinion, the most likely of these hypotheses is that when it is winter, everyone is indoors rather than outside, and when everyone is indoors, we're in much closer contact, and therefore we're far more likely to spread the influenza virus between us. Whereas when we're outside, if you cough and sneeze outside, the wind will just blow all of the influenza particles away and no one will breathe them in. So that's most likely the reason that influenza is so much more prevalent in the winter months, because everyone spends so much time indoors, whereas when you're outdoors, it's far more difficult to transfer the virus between people. Okay, so there's transmission. Let's now actually go on to what's going to happen. So, the healthy person has, let's say, breathed in loads of these influenza virus particles. What happens next? Well, most of them are going to end up in the nasal cavity, and indeed the nasal cavity is usually the place that first gets infected in influenza, and then it spreads down the respiratory tract, as we'll discuss. So in come loads of influenza virus particles, and most of them are going to get stuck in the mucus that lines the nasal cavity. So remember, the nasal cavity is coated by a respiratory epithelium, a ciliated pseudostratified columnar epithelium that has a layer of mucus over its surface. Most of the influenza virus particles, as they come in, are going to get stuck in this mucus, and of course what will happen is the cilia will beat and move the mucus backwards so that it can be swallowed, and then of course any influenza virus particles that are in that mucus go down to the stomach, and they do not like the stomach that much because the stomach has a pH of 1 or even worse, so they burn and burn and burn and they don't cause any problem at all. This is the reason that in order to actually get the disease, you have to breathe in quite a lot of the influenza virus particles, because most of them are going to be destroyed. If you breathe in a huge number, however, what ends up happening is a few of them will end up getting through the mucus to the underlying ciliated columnar epithelial cells, which are the target for infection. Some of them will get through the mucus down to there if you breathe in enough of them. If you just breathe in, let's say 10 or 1 even, then that's not going to give you influenza, because they'll all just get stuck in the mucus will end up in the stomach and they will be destroyed. You have to breathe in a lot so that eventually one of them ends up getting around our brilliant protective mechanism and getting to the underlying epithelial cell, and then it can begin its replication cycle. So, now what we want to discuss is the replication cycle. I want to discuss how is this virus particle of either influenza A, influenza B, influenza C going to get into this cell and going to use that cell to make loads more virus particles. We want to discuss then the viral replication cycle. So let's come back down here, put an evil red title of replication cycle, because this is the virus's evil mechanism by which it's going to replicate itself. And then let's begin discussing this. So, the replication cycle is going to occur in these ciliated columnar epithelial cells. So let's start by drawing a picture of a ciliated columnar epithelial cell. We'll have it here, like so. So at the top is the apical side of this cell, and I'll even draw a few little cilia on here. And then the bottom is the basolateral side, which will be sitting on the basement membrane. And of course, where is the virus particle going to arrive first? It's going to arrive on the apical surface of the cell. So the virus particle has somehow managed to get through the mucus, and now it's arriving at the apical surface of the cell. What happens next? It needs to first bind to something on the surface of the cell. That's the first thing it has to do. If it's going to get into the cell, it has to bind to something on the surface of the cell. What do the virus particles bind to? Well, this is where there is a difference between the influenza virus species. Influenza A and influenza B binds to a molecule, and here's the answer that you've been waiting for so long. Influenza A and B binds to a molecule called sialic acid, whilst influenza C virus binds to a slightly modified version of this. They bind to a molecule called 9-O-acetyl sialic acid. So very, very similar, but nevertheless slightly different. So what I now want to do is go over the structure of these molecules, and this will allow us to explore why the enzyme neuraminidase is called neuraminidase. It will also allow us to explore what the difference between these two molecules is. Okay, so let's start off with sialic acid then. So sialic acid, it's a modification of another molecule called neuraminic acid. So if we're going to discuss the structure of sialic acid, we firstly need to discuss the structure of a molecule called neuraminic acid. And this is now the reason that the enzyme neuraminidase is called neuraminidase, and why it has this other name of sialidase. So in actual fact, neuraminidase should be called sialidase because it is going to work on sialic acid molecules rather than neuraminic acid molecules. However, because sialic acid is a modification of neuraminic acid, the enzyme was called neuraminidase, and neuraminidase is still the more popular name for it over sialidase. However, sialidase is the slightly more correct name for it than neuraminidase. Okay, so the hemagglutinin molecules of influenza A and influenza B, and I'll just put this here. This is what the influenza A virus is, and the influenza B viruses are going to bind to using their hemagglutinin molecules. So those binding sites of the hemagglutinin molecules, they can bind to sialic acid molecules. This sialic acid is a modification of a molecule called neuraminic acid. So the first thing we're going to now discuss is the biochemical structure of neuraminic acid. Now, neuraminic acid is a monosaccharide. It's a sugar molecule, and like all sugar molecules, it's therefore complicated by the fact that it has a huge number of different optical isomers, which means that drawing a chemical structure of it is complicated. Now, the two important optical isomers that we need to know about are the fact that it has an alpha anomer and a beta anomer, and I'm going to show both of those structures. It's the alpha anomer that's going to be really important for hemagglutinin being able to bind to it, as we'll discuss. So let me now draw you out the structure then of neuraminic acid. So as they say, it's a monosaccharide, so it has the classical monosaccharide structure where you have the six-membered ring consisting of a single oxygen atom and then 5 carbon atoms, like so. And I'm drawing a skeletal structure here, so we're not going to show carbons, they're implicitly shown by corners, and we don't show hydrogens coming off carbons where you have missing bonds. It's assumed that those are to hydrogens. Okay, so we have 2 anomers then of neuraminic acid. I am going to start by drawing the one that is important to us, which is the alpha anomer of neuraminic acid. Okay, so we're going to start off with this carbon here then of the molecule, and it's going to have 2 important groups coming off it. And we're going to imagine that this, whoops, I meant to have the laser pointer at that point, let me just take that away. Give me the laser pointer. Right, so we're going to imagine that this 6-membered ring here is in the plane of the board, and therefore the other 2 groups that are coming off this carbon, one is going to be coming out of the plane towards us, and one is going to be going into the plane away from us. So one's coming out towards us, the other is going to be coming into the plane away from us. And the way that I show bonds coming out towards us is with a line that gets progressively thicker, and the way that I show bonds going into the page away from us is with a dashed line. Okay, so there are 2 groups coming off here. One's a carboxylic acid group, and one is an alcohol group. In the alpha anomer of neuraminic acid, the alcohol group is going to be going into the page away from us, so with a dashed line there, that means it's going into the page away from us. And then the carboxylic acid group is going to be coming out of the page towards us, and therefore has this line that's getting progressively thicker, like so. So this is the carboxylic acid group, like so. Okay, now, the carbon of the carboxylic acid group, this is numbered up as carbon number 1 of the neuraminic acid molecule, and then we go round. This is carbon number 2, this is carbon number 3. Now, carbon number 3 is a boring carbon. It actually just has 2 hydrogens coming off it, so we don't need to add anything onto carbon number 3. Carbon number 4 does have something interesting coming off it. It has an alcohol group coming off it, and then a hydrogen atom coming off it. So we need to know which orientation is this. Does the alcohol group go into the page, away from us, or come out of the board towards us? And I apologise for continually moving between page and board. If you are familiar with my channel, you will know that I used to do everything on paper rather than with this fancy electronic device, which is why I'm getting confused as to whether to say page or board. I apologise, I should be saying board. Okay, so on the 4th carbon, the alcohol group is going to go into the board away from us. So I'll draw a dashed line like so, and the hydrogen will be coming out of the board towards us. Then let's go to the 5th carbon. So around here, this is the 5th carbon. On the 5th carbon, you're going to have an amino group coming off this carbon, and it's going to be coming out of the page towards us, whilst the hydrogen coming off this carbon will be going into the page away from us. Now, this amino group is going to be important. We are going to stick an acetyl group onto this amino group in order to turn the alpha anomer of neuraminic acid into the alpha anomer of sialic acid. So this is going to be coming, it's going to be very important in the modification that's going to make sialic acid. Then we come up to carbon number 6 here, and carbon number 6 is going to have a free carbon chain coming off it, as well as a hydrogen atom. The hydrogen atom is coming out of the page towards us. The free carbon chain is going into the page away from us, so I'll show it with a dashed line like so. And then I'll put the other 2 carbons on here, so here's the other carbon, and here is the 3rd carbon. So those are the 3 carbons, and it's going into the page away from us, which is why I've drawn this line here, dashed. Now, it gets even more complicated, I'm afraid, because this carbon here and this carbon here also have alcohol groups coming off them, and they are also, therefore, chiral centres, and therefore we need to be aware of their optical isomerism. So let's go to this carbon here and ask which way is the alcohol group going to come, and the answer is it's going to come out of the page towards us. So if you imagine that this has gone into the board away from us, and then we've sort of oriented these 2 carbons, so that these lines are still in a parallel plane to the plane of the board. Yes, they're in a plane behind the plane of the board, but they're in a parallel plane. Then the alcohol group that comes off this one will be coming out of that plane back towards us, and the hydrogen will be going in away. So I'll draw this here. So here is this alcohol group coming out of the plane towards us, and then on this carbon here, the alcohol group is going into the page away from us, and this is where it gets awkward. How am I going to fit this in? I have to turn that into the alcohol group there. So the alcohol group off this one, again, that's a chiral center here. It's got 4 different groups coming off it. The alcohol group will be going out of the plane away from us, and the hydrogen will be coming out of that plane towards us. And then finally, just to make it even more complicated, off this 9th carbon over here, you've got an alcohol group. Now, thankfully, that isn't a chiral center, so we don't need to worry about the orientation. Okay, so just finishing the numbering up of the carbons, this is 7, this is 8, this is 9. So overall, neuraminic acid is a 9 carbon molecule. And the molecule that we have just drawn is the alpha anomer of neuraminic acid. So how would you turn this into what's called the beta anomer of neuraminic acid? Just for a bit of background knowledge, you would change the orientation of this carboxylic acid group coming off carbon number 2. Now, the way you decide whether a carbohydrate molecule is an alpha anomer or a beta anomer is you look at this group that's coming off this, this carbon of the 6-membered ring, and you look at the major group that's coming off this carbon. So you look at these 2 carbons that are on either side of the oxygen here, and you ask, what are their major groups? And the major group coming off this one is this, of course, not the hydrogen, and the major group coming off carbon 2 is this one. And if those 2 groups are going off in the same orientation, then we call it the beta anomer, and if, as is in this case, they're coming off on different orientations, which is true here. This carboxylic acid group is coming out of the board towards us, whilst this free carbon group is going into the board away from us. They're going in different orientations, then that's the alpha anomer. So there's a bit of background biochemistry. Okay, so to make the beta anomer of neuraminic acid, I'm not going to draw it all out. All you have to do is have the carboxylic acid group going away from us, into the board, away from us, and have the alcohol group coming out of the board towards us. Now, as I say, beta anomer is not important to us. The one that's going to be really important to us is the alpha anomer. So, just to make sure that you understand what I have drawn here, this is the alpha anomer of this molecule called neuraminic acid, which is important because we are going to modify it to make sialic acid, which is the thing that hemagglutinin of influenza A and influenza B molecules bind to. So, as I've already told you, let's now go over the modification that we make to neuraminic acid to turn it into sialic acid. So I spoiled the surprise. What you do is, to make sialic acid, you just stick an acetyl group onto the amino group that comes off the 5th carbon here. So another name for sialic acid, and I'll just find some interesting colour to do this in. Another name for sialic acid, and I'll go up here. Another name for this is to call it N-acetyl, because you're going to stick an acetyl group onto the N, the nitrogen atom of the amino group, and then of course it's just neuraminic acid, so I'll just put Na for neuraminic acid there, and therefore you might see sialic acid called NaNA, standing for N-acetyl neuraminic acid. Okay, another name for sialic acid. So, drawing this modification on here then, I don't know if I've really got space to do this in. What I'll do is I'll just draw the amino group out again down here, and I'll draw it modified. So, drawing this amino group out in more detail, here it is. Here's the nitrogen with the 2 hydrogens bound to it, and of course here is the bond that links it to the ring of the neuraminic acid molecule. Now, what are we going to do then? We're going to take off one of these hydrogens and we're going to bind the nitrogen instead to an acetyl group via an amide link. So let's just put this here. So here is the acetyl group. Here's a carbon double bound to an oxygen, and then all you've got is a methyl group bound to this, so H3C there. You're going to get rid of this, so that goes, and you're going to form a bond between the carbon and the nitrogen, which is an amide bond, and then you have an acetylated amino group there. So, when you do that to neuraminic acid, you end up with N-acetyl neuraminic acid, which is more commonly just called sialic acid. So this is the molecule that is going to be on the surface of cells and which the hemagglutinin molecules of influenza A viruses and influenza B viruses can bind to. Now, finally, let's come to this 9-O-acetyl sialic acid, which is the molecule that the influenza C virus hemagglutinin esterase fusion protein is capable of binding to. So how do we take sialic acid and turn it into 9-O-acetyl sialic acid? Well, hopefully, if your chemistry is good, well, not even good, if your chemistry is moderate, if you've got even slightest sort of chemical instinct, you will be able to work out exactly what you need to do. You need to stick an acetyl group onto an oxygen atom, and which oxygen atom do you do it onto? You do it onto the 9th one. You see, with the name N-acetyl neuraminic acid, you did not need to specify which N you were talking about because there was only one N in the entire molecule, whereas with oxygen there are loads of oxygen molecules, and therefore you have to put this 9 in front to specify which oxygen you are talking about. So here's the 9th carbon here, so we mean the alcohol group that comes off the 9th carbon, this one here. So what we need to do is we need to take that hydrogen off that oxygen, and just as we did with the nitrogen, we will attach one of these groups on there. So 9-O-acetyl sialic acid is neuraminic acid with 2 acetyl groups bound to it, 1 acetyl group attached to the amino group, and 1 acetyl group attached to the alcohol group. So we now know what these molecules, sialic acid and 9-O-acetyl sialic acid, actually are. Now, both of them use the alpha anomer of the neuraminic acid. So we're talking about the alpha anomer of both of these molecules. That's the important one for influenza A virus and influenza B virus hemagglutinin binding, and again, the alpha anomer of this is important for the influenza C virus hemagglutinin esterase fusion molecule binding. So, we now know that this is the target for these molecules, hemagglutinin and hemagglutinin esterase fusion, and I feel like I should put hemagglutinin here. I feel like it's unfair. Hemagglutinin esterase fusion got put over here, so I feel like hemagglutinin should be there. Okay, we've now talked about the fact that these are the targets, but what we need to question is, why are these molecules on the surface of cells? Where are they on the surface of cells? Have you ever heard about these molecules before? No, maybe not. Where are they on the surface of cells? And the answer is that they are a component of the oligosaccharide chains of glycoproteins and glycolipids that are in the cell membrane. So, let's draw a little picky. So, over here, these 2 lines can represent the phospholipid bilayer. Now, we know that the phospholipid bilayer is made up of loads of lipid molecules, loads of phospholipid molecules. We also know that in the phospholipid bilayer, there are lots of protein molecules. So I will draw one of these. So this molecule here in this bright red colour can represent a protein molecule. And I'll get some other interesting colour. Let's have bright purple. And this box here in purple, this can represent a lipid in the phospholipid bilayer that is a component of the phospholipid bilayer. It's helping to make the outer leaflet here of the phospholipid bilayer. Now, some of these proteins and lipids in the phospholipid bilayer have sugar molecules, sugar chains, oligosaccharides linked onto them. So let me find some other interesting colour. We'll have the aquamarine colour. So some of the lipids and proteins in the phospholipid bilayer have oligosaccharides linked onto them. Now, what does oligosaccharide mean? Well, we know what monosaccharide means. That means a single component of a polysaccharide, a single sugar molecule. A polysaccharide means a great big long polymer of monosaccharides joined together. So things like starch and glycogen are polysaccharides. Oligosaccharide is something in between. It's a very short polysaccharide, effectively. Oligo literally means some, whereas poly means many. So when you were talking about a polysaccharide, that means you've got loads and loads of monosaccharides joined together in a great big polymer. When we're talking about oligosaccharide, that means you've got some of the monosaccharides joined together in a chain. It's not as long as a polysaccharide. So oligosaccharides are kind of 10 to 50 monosaccharides long, whereas polysaccharides might be hundreds and hundreds of monosaccharides long. Now, so why am I talking about this? Well, as I say, these protein molecules and these lipid molecules, some of them can have oligosaccharides linked onto them, like so. So these lines are now representing these chains of monosaccharides that we're calling oligosaccharides, and they might not just be linear chains, they might be more like tree structures, like this, because of course monosaccharides are complex. You can link other monosaccharides in many different ways to a single monosaccharide, and therefore you can get branching of these monosaccharide chains. So I might just draw a few more on here. So here's another one of these oligosaccharide chains attached to this protein. And when you have a protein that has lots of oligosaccharide chains bound onto it in this way, it's called a glycoprotein. So glyco means sugar. So these are proteins with sugar modifications on them. Now, not only can you do it to proteins, but you can also do it to lipid molecules. So this lipid here may well have oligosaccharide chains linked onto it as well, just like this. And when you have a lipid molecule that has sugar modifications attached onto it, what do you think that's going to be called? This is not difficult. They're called glycolipids. So, in the cell membrane then, you have a lot of glycoproteins and you have a lot of glycolipids. Now, why is this relevant? Because sialic acid molecules and 9-O-acetyl sialic acid molecules can be components of these oligosaccharides of glycoproteins and glycolipids. In particular, the ones that the hemagglutinin molecules of influenza A virus and influenza B virus are capable of binding to will be very terminal sialic acid molecules. So, let me just get the dark blue pen back. So, some of these oligosaccharide chains of these glycoproteins and glycolipids that coat the cell surface will have at their end, and here it is in blue, a sialic acid residue. Oops, I've gone to the thicker pen, but never mind, it's still usable. So some of them will have right at the very end a sialic acid residue, which is what I'm trying to communicate with this blue dot. Right at the end of that chain, the terminal residue there is a sialic acid residue. Not all of them, of course. This one might not have a sialic acid residue as the terminal, but many of them do have sialic acid residues as the terminal one. In addition, you'll also find that some of them will have 9-O-acetyl sialic acid residues at their terminals. So maybe we'll have this down here in green, and maybe I'll put another one there. So this might be 9-O-acetyl, and then I'll just use the abbreviation for sialic acid, NA, like so, N-acetyl neurominic acid. So, my point here is that on the surface of the cell, you have lots of glycoproteins and glycolipids, which have these oligosaccharide chains. Now, some of those oligosaccharide chains, at the very terminal residue position, will have a sialic acid residue or a 9-O-acetyl sialic acid residue, and these are the sialic acid and the 9-O-acetyl sialic acid molecules that hemagglutinin of influenza A virus and influenza B virus and the hemagglutinin esterase fusion molecule of influenza C virus can bind to respectively. So to make that clearer, influenza A and B can bind to the host cell surface by binding, using their hemagglutinin molecules to bind to these terminal sialic acid residues of the oligosaccharides of glycoproteins and glycolipids, whereas influenza C virus can use its hemagglutinin esterase fusion protein to bind to 9-O-acetyl sialic acid molecules that are on the terminal of these oligosaccharides of glycoproteins and glycolipids. And remember, each of those molecules, hemagglutinin and hemagglutinin esterase fusion, they are trimers. They have 3 binding sites, and each of those separate binding sites can bind to a separate one of these molecules. Okay, right. So that is how the influenza viruses bind to the surface of the cell using hemagglutinin and hemagglutinin esterase fusion for the respectively binding to these molecules. Okay, so we get the influenza A or influenza B or influenza C virus binding to the surface of the cell. Now, on this picture, I will just draw the virus as an evil red dot. So the bit that we've discussed now is the binding of the virus like so. So we have gone over binding. We understand that hopefully now completely. That's the first thing that happens. Once it is bound to whatever molecule it does bind to on the surface of the cell membrane, it's then going to be internalized by endocytosis. Now, there are lots of different mechanisms of endocytosis, and it is not, thankfully, understood which of these influenza viruses use. It's still very controversial, and therefore I can skip it out. I don't need to go into the details of the mechanisms of endocytosis, so we will skip this bit out. We've got enough to discuss in this video without going into the detailed mechanisms of endocytosis. So instead I'll just give you some general comments on endocytosis once I've got the pen back. So what's going to happen next is this process called endocytosis. Now, endocytosis is the process where a portion of the cell membrane is internalized to form an internal membrane-bound cavity, and that's it. That's what endocytosis means. The internalization of plasma membrane, cell membrane, to form an internal membrane compartment, which is called usually an endocytic vesicle. So, the influenza virus binds to the surface of the cell membrane, and then what's going to happen is it's going to be endocytosed, so the membrane that it's bound to will be internalized, and it will form an endocytic vesicle that's going to contain the influenza virus. So let's draw this on here. So I'll draw it here. So in green, this is now an endocytic vesicle here. It's an internal portion of membrane that has come from the plasma membrane, so it's been pinched off from the plasma membrane, and now the influenza virus is going to be inside that endocytic vesicle because it's still bound to the molecules that were in the membrane, the sialic acid and 9-O-acetyl sialic acid molecules that are now in the endocytic vesicle. So all of the glycoproteins and glycolipids that it was bound to. And I should stress, when an influenza virus binds to the surface of the cell, it will not be linked just by a single sialic acid residue. It's not like a single hemagglutinin uses one of its binding sites to bind to a single sialic acid residue. No, that's not the way it works. You've got loads of hemagglutinin molecules and loads of hemagglutinin esterase fusion proteins if you're an influenza C virus, and loads of them all at once will bind to multiple different target ligand molecules on the surface of the cell. So you get a huge amount of binding. And all of those glycoproteins and glycolipids, they'll end up in the membrane of the endocytic vesicle after we've had this internalization occur. Now, as I said earlier, there are lots of different mechanisms by which you can pinch off a portion of the plasma membrane and turn it into an endocytic vesicle like this. The famous ones are clathrin-mediated endocytosis, caveolin-mediated endocytosis, macropinocytosis. However, it's still controversial which one of those the influenza virus actually uses, so we won't go into it. We'll just have this nice simple picture here of it being endocytosed. We don't need to go into all the molecular machinery that mediates that. Okay, right. So, we're now at the stage where we have the influenza virus inside our endocytic vesicle here. What now happens to endocytic vesicles? Well, they go into the endocytic pathway, and I will give you a little bit of the background about the endocytic pathway so that you understand what's actually happening here, and I need to change the pen thickness again. Okay, so endocytic vesicles enter what is called the endocytic pathway. So, what is the endocytic pathway? Well, what happens is loads of the endocytic vesicles that are coming from the plasma membrane all the time. So endocytosis is occurring all the time. The cell uses it all the time. So you're getting a lot of endocytic vesicles produced. What happens is loads of endocytic vesicles fuse together to make a much bigger membrane-bound compartment called an early endosome. which I'll show. And I'll just try and give it a few little tubular parts here on the picture. Okay, this is going to be my picture then of the early endosome. So this structure here in purple, this is the early endosome. So what will happen is our endocytic vesicle that is coming with the influenza virus inside it will come and fuse with this early endosome, and its membrane will now contribute to the early endosome, and that means that the influenza virus particle will end up in the lumen of the early endosome. Now, before we actually discuss what will happen to the influenza virus in the early endosome, I just want to discuss what's the normal purpose of endosomes. What's the normal purpose of the endocytic pathway? Well, what do cells normally endocytose into endocytic vesicles? Normally, it is nutrient particles. The purpose of endocytosis is that all sorts of nutrient particles that the cell would like to use and break down to get nutrients can be arriving at the surface of the cell. What happens to these nutrient particles? And for the meantime, just imagine that this red dot is not representing an influenza virus. Instead, it's representing some lovely nutrient particle. You could imagine that it's a lipoprotein, potentially. LDL is the classical example, which delivers us some beautiful nutrients. I know LDL has a very bad reputation in the tabloids. It's called bad cholesterol. But in reality, it delivers to cells. And the reason it's got such a bad reputation is if you have too much of it in the blood, it leads to atherosclerosis, which leads to heart attacks and strokes. So it is bad if you have too much of it, but too much of a good thing is always bad. So, LDL, actually, the function of it in the body is to deliver to all the cells of the body cholesterol. So let's imagine that this is a nutrient particle. You could be thinking LDL as a typical example. What happens is it binds to some receptor on the surface of the cell, and in the case of LDL, there's a receptor called the LDL receptor. Then that receptor mediates its endocytosis, so you get the nutrient particle endocytosed, and it will still be bound to its receptor that will also be in the endocytic vesicle. This is then brought to the early endosome, and the endocytic pathway, which is all about these endosomes, is all about trying to break down the nutrient particle to release the nutrients. So in the case of LDL, to release the cholesterol so that the cell can then use the cholesterol, and it uses it to make cell membrane. Okay, so in the early endosome, what will happen is you'll end up with the nutrient particle, and I will just draw this. I will draw the virus particle arriving in the endosome. So here is our nutrient particle, which at the moment it's representing a nutrient particle. It's arrived in the early endosome, but it's still at the moment bound to the receptor. Now, what's going to happen is this early endosome gradually matures and matures and matures and turns into something called a late endosome, which I'll draw here, which doesn't have as interesting shape. It doesn't have these interesting projections, which I'll tell you the purpose of in just a moment. So in green, this is representing a late endosome. So the endosome gradually matures. When it gets big enough, it gradually matures into something called a late endosome, and this changes its shape. So it turns into this thing. And what will happen is when the late endosome has finished its maturation process, it will fuse with lysosomes. So let me just draw some lysosomes in this pale green here. This can represent a lysosome. Now, lysosomes are these membrane-bound compartments inside cells that are full of horrible enzymes. Useful enzymes, but horrible enzymes in the world of molecular biology. Enzymes that break down all sorts of biological molecules. You'll have enzymes that break down DNA, enzymes that break down RNA, enzymes that break down lipids, enzymes that break down proteins. They are filled with enzymes that we can overall describe as lytic enzymes. So lytic means to break down. These are enzymes which break down biological molecules into their component pieces that can then be used by the cell and to produce new biological molecules. So, remember the function of the endocytic pathway. We are bringing in nutrient particles. Those nutrient particles are going to remain in the endosome as it matures into the late endosome, and then lysosomes are going to fuse here and the nutrient particle will be broken down. So if this is LDL, it will end up here and it will be broken down by the lysosomes, and we'll get loads of useful nutrient molecules from that the cell can then use. That is the function of the endocytic pathway. Now, there's a problem here, which is we have brought in this nutrient particle by it binding to some receptor. In the example, LDL binds to the LDL receptor. If the receptor was to remain in the endosome as it matures, the receptor would be destroyed when the lysosomes fuse with the late endosome as well. And that's not very efficient. We don't really want our beautiful receptor that is so useful to be destroyed. We would rather it could be recycled to the plasma membrane to perform this role of bringing in the nutrient particle again. And that is indeed what happens. So, in the endosome, as it matures, the pH gradually gets lower and lower. So this is why I'm telling you this entire story, to justify why the pH gets lower and lower in endosomes. So as the endosome matures, the pH gets lower and lower, which remember means it's getting more and more acidic. The proton concentration is going up and up. Now, what's the purpose of this? When proton concentration goes up and up, it causes conformational changes in proteins. And the idea is that these conformational changes in the receptor that is bound to the nutrient particle will cause the nutrient particle to detach from its receptor, and then that we can recycle the receptor back to the plasma membrane, which is what these tubular portions on the side of the early endosome are. These are places where you can bud off little vesicles, and I'll just get another little colour for this. We'll have it in not evil red. So, this is a little vesicle that has budded off that tubular process of the early endosome. So it's broken off there, and it's now going to deliver things back to the plasma membrane, and that's called a recycling vesicle. So the idea is then, to summarize the entire endocytic pathway here, the idea is that here is some nutrient particle that we want to use, this cell wants to use. What will happen is it will bind to some receptor on the surface of the cell, which will mediate the endocytosis, so the nutrient particle will nicely end up inside this endocytic vesicle. Fantastic. The endocytic vesicle will then fuse with this early endosome, which is formed from loads of endocytic vesicles joining together. Its membrane will then become part of the early endosome, and what you'll now have in the membrane of the early endosome is the nutrient particle bound to the receptor. The receptor is now in the membrane of the early endosome. What now happens is, as the endosome matures, the pH gets lower and lower and lower, and the idea of this, the function of this, is so that the protein here, the receptor, can change conformation, detach from the nutrient particle. The nutrient particle can float off into the lumen of the early endosome. Then what can happen is the receptor can go into one of these tubular portions of the side of the early endosome and then end up in a recycling vesicle. So you'll get the budding off these recycling vesicles, and this recycling vesicle can take the receptor back to the plasma membrane. Fantastic, we recycle the receptor, whilst the nutrient particle stays in the lumen of the early endosome. The endosome matures into a late endosome, lysosomes fuse with the late endosome and will break down the nutrient particle that is still in the lumen of the endosome. Okay, now as maturation continues, the recycling process stops, so the tubular portions on the side of the endosome disappear. So that's the endocytic pathway. That's normally how it functions. The influenza viruses hijack the endocytic pathway. So I wanted to just go over that little bit of background so that you understand how it normally works, why it is there. You understand the normal physiology before we now discuss the pathology, which is hijacking it. So, this is no longer a nutrient particle, this is something far worse. This is a influenza virus. So it's been endocytosed, which is mediated by the sialic acid or the 9-O-acetyl sialic acid molecules. It ends up then in the early endosome, and what's going to happen in the early endosome is the pH is going to go down and down and down. Now, you should remember something that I told you about what happens when the pH around an influenza virus goes down and down and down. It's going to activate the M2 ion channel, and the M2 ion channel is then going to conduct protons into the inside of the virus. Okay, I think we will have a break here and we will continue on this story in the next video, and we'll see how the virus is going to escape from this early endosome, which of course it wants to do. It doesn't want to stay in here. If it stays in here, it's doomed. It's going to be broken down like everything else. So there's an escape mechanism that's coming up, and this is going to involve hemagglutinin or hemagglutinin esterase fusion, and also the M2 ion channel. But we'll see that in the next video.